This is a production of Cornell University.
I want to thank the committee and Alan for this opportunity to give an update on some of the things that we have going on with compatibility for fungicides and insecticide seed treatments with rhizobia and biocontrol agents. I think this is a really interesting topic. And a lot of times when I'm asked to give a presentation, these type of talks to groups like this are really interesting to me because it's a topic that's a little bit different than I'm normally used to talking to, which is more about how our products work and efficacy and things like that. But when I give one of these talks, I do quite a bit of research and I tend to learn a lot about this new topic. And it was in this case too that I really felt like I was able to learn some things. So I hope that through this time that we spend here in the next few minutes, I can give you some of that information that I learned and that you'll find it interesting. So rhizobia are important in legume crops for increased nodulation and nitrogen management. Their use decreases the need for synthetic fertilizers and helps growers farm more sustainably. Rhizobia products are often seed applied, and the advances in seed applied biocontrol agents have also occurred in recent years, and many new products are being brought to the market and are often seed applied as well. Fungicide and insecticide seed treatments are still very valuable to growers, and the compatibility of these different products are very important to ensure that growers are getting the expected benefit of all the products applied. Rhizobia and biocontrol compatibility begins with the design of the fungicide or insecticide formulation and includes the active ingredient as well as the choice of inert ingredients. The compatibility is evaluated at several stages, which I'll go through the development process and is often checked by rhizobia product companies as well before commercialization. And also responsible biocontrol product companies are also testing for compatibility of their products when it makes sense and they believe that their product is going to be applied as a seed treatment. And by working together, the industry is really trying hard to make sure that these products are working together and can be used effectively to the best maximize the yield and farm sustainability. So that's in a summary really what I want to share with you today, and I'm going to go into a lot more detail now about these. So as you know, Rhizobium is a genus of soil bacteria, and the fact that it's bacteria means that this is a living organism that's being applied to the seed, and that brings certain considerations that we need to think about. An important feature of these Rhizobium is that they're very specific to the legume or that they're being applied to. So some of them are only specific to a very small amount of the crops. For example, some of them are only nodulate on alfalfa species, whereas other Rhizobium can be very broad spectrum across a number of crops. For us, there's Rhizobium leguminosarum nodulates on pea crops, and Brady Rhizobium japonicum nodulates in soybeans. This is important because we're not just talking about one specific bacterial organism that is applied across all crops that we need to worry about, but there's many different Rhizobia species that we have to think about and test for when we're talking about compatibility with seed treatments that are also going on that same space on the seed. And I just have a picture here of, you can see the nodulation on the roots. So what we're talking about is these Rhizobia that are applied to the seed and then, or in furrow in some cases, and then cause these nodulations. And what this is really doing is this Rhizobia bacteria is colonizing the plant cells within root nodules, and they convert atmospheric nitrogen into ammonia, causing the enzyme nitrogenase, and then provide organic nitrogenous compounds such as glutamine or urides to the plant. So it's really helping the plant. The plant in turn is providing the bacteria with organic compounds made by photosynthesis. And this is a mutually beneficial relationship, and it's true of rhizobia, which is of which the genus Rhizobium is a typical example. So this mutually beneficial relationship is something that we really want to make sure we're protecting and make sure that we're taking advantage of, especially when we're trying to reduce fertilizer use, synthetic fertilizer use as much as possible. And we want to try to increase our soil health. So rhizobium can be formulated with other microorganisms to improve or add additional benefits. And I've just taken a snip here from Novozymes document, and you can see that there's a Rhizobium leguminosarum, but there's also penicillium on here as well. And these Rhizobium products can actually take different carriers or different forms. You can have peat, you can have granulars, or you could have liquid inoculants. So all of these need to be considered as well. So that's a little bit about the Rhizobium, but what about a seed treatment? And although it sometimes might seem like it, you just had a nice talk from Peter Marks, and you can make these seeds look really nice, and it might seem like it's about the colors and the shiny appearance. It's more about what the products are doing to protect them against insects, nematodes, and diseases, which are mainly fungi. And these have been very useful to growers, and they're getting a lot of benefit out of them. You can see here, if you have an untreated seeds and you have planted in the presence of Pythium, sometimes you'll just basically get no plants, but a treated seed can protect you from Pythium. And this can be demonstrated in little rhizotrons like this, where we've inoculated the soil. We can also do Petri plate studies. You can see here an untreated Petri plate with Rhizoctonia in the center and the Rhizoctonia mycelium growing out across the seeds, whereas the treated seeds are protected from the Rhizoctonia. And this is the benefit that these seed treatments are giving to growers. So we want to be able to combine both of these technologies together and be able to use them efficiently. We want them both to perform well. When we're talking about the pesticide formulation, there is the pesticide active ingredient. That is the main component that we're talking about. That's the thing that's going to be controlling the pest. There's carriers, water or organic solvents that are also in those formulations. These are all the inert or other ingredients. And when we're talking about compatibility with something like a Rhizobium or bacteria, we really need to consider all of these inert ingredients as well as the active ingredient. Other things that can be in there are surfactants or adjuvants, stabilizers. You can have dyes or colorants and safeners, as well as antifreezes to keep it from freezing in cold temperatures. Another thing that is often or almost always put into a pesticide formulation or a seed treatment formulation are synthetic biocides. So the synthetic biocides are placed in there so we don't get bacterial contamination growing in the jug. If you've ever opened a jug and it smells really bad, you know you have some bacterial contamination and the biocides weren't doing their job. So these are all components of a seed treatment formulation. And you can see that some of these are going to cause some problems when you're trying to be compatible with an organic or with an organism that's a living. Organic solvents, biocides could be an issue. And so we need to take care of that. And how are we going to do that? I just want to say, Syngenta has a long history of developing these things. We've been doing this for over 40 years, and we spend a lot of money and time investing in our formulations, nearly $4 million a day. And we don't want to have a product that is going to come to the market and not be successful or not be able to be used. because of its incompatibility with a rhizobium or another biocontrol product that our growers really want to apply. To help us with this, we do have dedicated seed care specialists around the country that listen to growers and try to understand how our products are working. And there also can be resources for you if you're having an issue with one of our products or have questions about them. You can always contact one of them. We have a facility in Minnesota, which is a Seedcare Institute, where we do a lot of training. But this is also where we do application testing, like Peter was talking about. And we have inoculum testing program as well as our seed safety testing program. So what does Syngenta do to keep our products compatible with inoculants? Actually, in the formulation development of a new seed treatment, we do tests for compatibility. And these tests are primarily to put the product on the seed after, we treat the seed, and then we put the inoculum on top, and then we'll see how long those inoculum last on the seed or how long it survives on the seed. And we have a certain threshold that we want to maintain for the number of bacteria that are still alive on the seed after a certain amount of time. And so we do test that in the development process. We only test a few of those to make sure that this product's not a significant issue. And if it passes that test, then we'll continue with the development of that formulation, and we feel that it'll be successful. We'll go ahead and freeze that formulation and commercialize it. However, after the product's frozen, or we've got a good idea of what that product's going to be, we've gone through some tests, we will offer our products for testing to other inoculant manufacturers for testing prior to commercialization. And if we come back and there's some big issues, we will withhold that product from the marketplace. Currently, the only company that's really been taking us up on this offer is Novozymes. So I'm going to be sharing some information from Novozymes. But they do test the compatibility of all our new formulations and treatments with their inoculant products. And it's something that Syngenta doesn't really have the expertise to do in-house. So we have the inoculant companies are really good at testing those things, and we have them do that. And then they give us back the information. And then they publish it in a guide. So you can download this guide I did last week from Novozymes. And it's a compatibility guide, and it gives you by crop all of the different products that they have. I've just taken a snip of one of them, which is Tag Team Pete, and then it lists all of the products that have been tested by Syngenta, or tested by Novozymes, I'm sorry. And you can see there's a pretty big list of those products here, and I just want to highlight a few. This one is a Apron Max RFC. The RFC actually stood for Rhizobium Friendly Concentrate. We did make some modifications to the formulation to help it be more friendly to Rhizobium actually than the original Apron Max RTA formulation. And you can see that it did seem to improve the planting window. And this planting window is actually the maximum amount of time that can elapse between the date the seed was treated with a bioenhancer or Rhizobium, and when it can be planted with an expectation that the Rhizobium will remain viable. And generally this is, I've seen these, really the maximum here is 48 hours on all of these tests that they do. However, we know from some of our internal testing that they do last on the seed, or they can last on the seed longer, but generally the greatest limit here is 48 hours. There are some products, older products, sometimes, or other formulations that you'll see are not recommended at all. For whatever reason, they were not compatible with the inoculum product. And so those are ones that you really need to look at and watch out for, and maybe choose a different inoculant that was more compatible with the product that you want to apply. Or potentially choose a different seed treatment product. Like I said, we do testing. We offer our products for testing. So these are products that aren't on the list or on the guide yet. So these are new Syngenta products that have been recently commercialized, and we've done testing with those. And you can see that they're also 48 hours for the simultaneous, sequential, and tank mixed uh, timings, application timings. And if you don't see one of the products on the application guide from the inoculant company, the manufacturer of the seed treatment may have this information with them. And we do put this information in our technical bulletins and technical presentations, so it is available, because we know that those guides aren't updated all the time. So how do we avoid problems with the seed treatment and inoculants? We want to avoid using products with negative effects. Of course, in the formulation process, we're trying to use the products that would be trying to avoid the ones that are really negative. And there's always the acute toxicity, the exposure dose, and the length of exposure. And if you see this lady girl sitting here, this is a famous painting from 1800s, and there's a really nice green wallpaper in the background. And she's lucky that she has the window open here, because this green wallpaper in the Victorian eras was color was due to arsenic, and they used arsenic to make that green color in the wallpaper. And they had a lot of problems with people actually getting sick when in the winter it was closed up, and they would be in here and actually get sick from the arsenic in the wallpaper that was coming out. So if she's lucky she has the window open. If she'd been outside or away from that, she could have avoided it or taken down the wallpaper, eliminated that. Then you're able to not be exposed to that toxicity. So in similarly, to test compatibility, we have different ways we can apply the inoculum or the biocontrol agent. You can do a simultaneous application, a sequential application, or a tank mix application. Tank mix application is kind of like being right in the room with the product. This is a diagram that I got from a BSF document from Canada, and it shows very nicely what a sequential application is, where the seed treatment must be applied and allowed to dry before the inoculant is applied. The simultaneous application, the seed treatment can be applied at the same time as the inoculant, but separate applicators must be used. And then the tank mix application, basically the inoculant is mixed directly with the diluted seed treatment and applied as one solution. However, they do recommend that the maximum hours of the slurry that's mixed up, and you have to be applied within 6 hours, and some other people recommend within 4 hours. There is also a best practices guide. So Novozymes has produced a bioag best practices guide for using inoculants, and it has a lot of really good information around how to keep these organisms working in compatibility with seed treatments. For living organisms, you know that they depend on us as applicators to exercise good storage practices before and after the application. You want to make sure that there's cool temperatures that favor the survival of the bacteria found in the inoculants. Store the products and seed to which the products have been applied in a cool, dry place, and refer to the conditions on the product and seed labels for specific recommendations on how to best keep those organisms happy. The other thing is you can have a dedicated tank and for the inoculum and a pumping system. This will allow for easy management of the inoculant or biological products. And while the standard seed treatments are generally stable, if left in a mixed tank for a few days before consumed, it's not true for inoculants. A dedicated tank would allow you to open and mix only what you need for immediate use and keeping your inoculant source as fresh and as viable as possible. And even with a dedicated tank, you need to apply all of the product within 24 hours after opening. And if you don't have a dedicated tank, you can choose to mix the inoculants with the treatment. But if this is done, that, like I said, you need to be using all of that slurry up within 4 to 6 hours. And another important thing is to clean out your equipment and make sure that you have good sanitation throughout so that the seed treatment and the inoculant that you use in the next batch are also compatible, or make sure things are clean. Another thing that a lot of people don't really think about is the water. So you may be treating with a city water, and as we know, they treat city water with chlorine to help kill microbes in the water. So this is not going to be compatible with your inoculum. And so if you're treating with the city water, which has chlorine added, you may want to install a water filter, which is designed to remove the chlorine, or use a spare tank where you can hold the water for 24 hours and allow the chlorine to dissipate before you use it in your slurry. Also, many treaters use soybean oil to help, or other oils to help with the treatment process. And generally, this has been shown not to be an issue and doesn't seem to have any effect or harm on the bacteria. And it also may even improve the quality a little bit because it dries a little bit slower than just straight water. Uh, it's also important to check all of the other things that you're adding to the mixture, the slurry. In addition to the seed treatment part, you could add other polymers or colors, or maybe another product from somewhere. And you want to make sure you check all of those when you're applying it with an inoculant. So important to think about all of the things that are going into your slurry. I've talked a lot about the inoculants. I've talked about the uh, seed treatments, and now another growing area is really biologicals. So a lot of these biologicals could be living organisms as well. What are really growers looking for a biological? We're trying to unlock soil-bound nutrients. We're trying to enhance access to those nutrients, fix atmospheric nitrogen, stimulate development, or protect against pathogens. And some of these can be live organisms or plant extracts, small peptides, all kinds of things that you want to make sure the integrity of those is maintained when you're mixing them. For biocontrol, usually they can be viruses, bacterial, fungal pathogens, pheromones or botanicals or other substances. Uh, in biostimulants, you could have microbial products, nitrogen fixing bacteria, algal extracts, humic acids, amino acids, peptides, enzymes, vitamins, all kinds of things. Nutrients are usually micro or macronutrients that generally are not considered incompatible with seed treatments in a living sense, but could be incompatible from a physical chemistry type of sense. So if the biological or the organism is actually a living organism, the same kind of rules would apply as we've talked about for the Rhizobium bacteria. You want to make sure that those are happy and those conditions for that organism are satisfied, and want to test and make sure that those bacteria are still alive on the seed after application, that it's compatible. And the testing for this compatibility is done by a lot of the biological companies that are bringing these new products to the market. We're often approached by companies that ask for product to test when they're starting out, trying to determine if they can apply a product to the seed and if that would be a mechanism of delivery for their product, or potentially they would have to go in furrow and do an application that way instead of applying it with the seed treatment. they're also trying to figure out whether they can do the tank mix, or it could be built into a formulation, a seed treatment formulation, or if it's better doing a sequential application, something like that. And so there's a lot of work that's been done in this area, but if you're not sure about the products you're using, or if they've been tested, it's always good to ask the manufacturer what the compatibility is and what kind of testing's been done. Syngenta doesn't do a lot of compatibility testing of all the products, the biological products on the market, unless we have an agreement with that company that we're interested in their biological, or they've asked us to test it, or um, asked us to supply some product for them to test. But there is testing going on. So I did mention, just a lot about the biologicals that we're thinking about and how they're compatible, trying to keep them alive. But there's also physical compatibility. And some of these other products that I mentioned, the humic acids, they're not living, but they're products that are being applied on the seed as biologicals. And it's important to look at the compatibility of those if you're going to apply those at the same time or put them into your slurry. And one of the best ways to do that is to do a jar test, and you can learn a lot from a jar test. That'll save you a lot of trouble cleaning out lines and tanks and cleaning up a mess later. We do tests with our products with biologicals that we are thinking about, or we know that may be put into a tank mix. And one of the things we're looking for is the incompatibility, just physical incompatibility, change in viscosity, change in appearance or physical behavior, or separation, like you can see here in this jar. And that's something you want to try to avoid. Sometimes there's a little bit of separation, but if it's recirculated easily, then we usually say that would be acceptable. There's also compatibilities where we increase the temperature, decrease the temperature, make sure that in a range of temperatures it's okay. So there's a lot of tests that are done with just physical compatibility. Also, it's very important to clean the equipment when changing formulation types. Just good cleanliness really helps out with seed treating and making sure that everything is compatible and flowing easily. And one way to clean out easily is using a simple green, something like that. So I did go through quickly quite a lot of information, and I just wanted to talk a little bit about some of the sources of the information I have. There's the Novozymes BioAg Seed Treatment Compatibility Guide. I did take some information from the 2013 one, but there is another one that was updated more recently, and there's a best practices guide that's available online from this January. There was some information you can find from compatibility of different inoculants from BSF in Canada. There's a Syngenta technical bulletins or technical bulletins from your company or other seed treatment companies about the inoculum compatibility or biological compatibilities. And then there's different papers that you can read about online, quite a lot of journal articles and other information that you can go to really dig into some of this as well. So with that, if there's any time left, I'd like to take some questions.
David, thank you very much for the talk. Yes, just a question or so. It's again packing a lot of information in a short amount of time. And again, yes, also again the CCA credits. There's your information to go ahead and get those CCA credits. So a question for David on compatibility of the seed treatment, chemical seed treatments with the rhizobia or the biocontrols. Okay, and I don't see anything in the chat right now. Okay, David, again, thank you very much. Thank you very much for this great presentation.
You're welcome.
This has been a production of Cornell University, on the web at cornell.edu.